Welcome back, class. So you have just finished your third exam. I hope you did very well on it. Usually at this point in class, you've kind of learned how I write tests and how they work, and hopefully you've gotten that down. These last few chapters, this last unit of class, we are going to take a deeper dive into the different types of microbes we've been studying this year and some of the diseases that they've been causing. This is a pretty clinically focused last few chapters, and so I hope it'll be pretty interesting to you. So our prokaryotes are divided into those with a peptidoglycan signature and a pseudomurine signature. So what has a RNA for peptidoglycan, what has RNA for pseudomurine. Those that do pseudomurine are going to be within the domain archaea. We'll touch on them very briefly at the end of this chapter. And those with peptidoglycan are divided into bacteria. Okay, so that's already pretty comfortable for you. Our bacteria are divided into gram-positive and gram-negative. Also pretty much a review for you. Then our gram-negative bacteria are divided into the proteobacteria. These are mainly chemoheterotrophic bacteria. And the non-proteobacteria, which is kind of the odds and ends that didn't fit into the proteobacteria. Then our gram-positives are divided by low G plus C and high G plus C. So I want you to stop and think for a minute, what could it mean to be low G plus C or high G plus C? And while you do, I'm going to come talk about Bergey's Manual of Systematic Bacteriology. So this is the reference source that classifies bacteria. You could actually go buy this manual. It goes through bacteria and sorts them by the ribosomal RNA. So we know all bacteria have a common ancestor, and the more closely related two species are, the more closely related those ribosomal RNA fragments will be. And as they get more and more, accumulating more and more mutations between each other, the more different their RNA is going to be, the more distantly related they're going to be. RNA is a preferred method really to classify any type of life because all life does have RNA. Although in bacteria specifically, it's very impossible to classify them by morphology because you can't see them. Okay, so now that we talked about how we classify them by their RNA, low G plus C and high G plus C, maybe you figured out we're talking about RNA nucleotides. So some have a low level of G's and C's, they have lots of A's and T's, and some have a high level of G's and C's, they have a lower level of A's and T's. So our gram-positive bacteria, the low G and C bacteria are the Firmicutes or the Firmicutes, and the high G and C are the Actinobacteria. Within the Firmicutes, we have Clostridium, Bacillus, Staphylococcus, Lactobacillus, Listeria, Streptococcus, Enterococcus, and Mycoplasma. We are going to go through and talk about each of these species, each of these genus, and then within the Actinobacteria, our high G plus C ratio, we have Mycobacteria, Corynebacteria, Streptomyces, Actinomyces, and Nocardia. So start off with Clostridium. These are rod-shaped cells that form endospores. We've discussed endospores before, so you should already know Clostridium form endospores. These are obligate anaerobes. So that means that they will be killed where oxygen is present and only grow where oxygen is not present. Within our Clostridia, we have tetani, C. tetani, which causes tetanus. And if you think about that wound or think about that infection, it's going to grow deep within a wound. It's not just if you lightly scratch yourself with a rusty nail, you're going to get tetanus on there. It's if you have a deep puncture, right? So where there would be no oxygen growing. C. botulinum causes botulism. Also, this is a food poisoning. It grows in improperly canned foods. So once that can is sealed up, if the endospores have not been properly killed, once the can is sealed, it's not getting oxygen. It's a great place for botulism to grow. As botulism grows, it'll typically start fermenting carbohydrates, which causes the volume inside the can to swell. So if you ever have a can that's just bulging, that is a can that would be a good choice to throw away because that might indicate that botulism has been growing in it. And then Clostridium perfringens, which causes gas gangrene. That's what's in this picture here. that will be a bacteria that will be fermenting the carbohydrates in the tissue and making gas bubbles within the skin, within your tissues. C. tetani causes tetanus. That's this picture here. And C. botulinum, which causes botulism. Let's talk about them a little bit more. So Clostridium tetani produces a toxin called a neurotoxin. So that tells you it's going to be related to affecting your nervous system. Sometimes it's also called tetanoplazmin. Neurotoxin will block the relaxation of your muscles. So you can see in this picture, his hands are clenched. Every muscle in his back is clenched. He's got this curve. This curving of his body is called opisthotonos. You can see his toes are clenched. Sometimes tetanus is called lockjaw. You can see in the picture his jaw muscles are clenched tight. So every single muscle in his body is unable to relax. It all tenses up. Botulinum causes creates an exotoxin that does the opposite. It blocks acetylcholine, which is an important neurotransmitter, so you end up with a flaccid paralysis. So here in this picture, you can see he can't even open his eyes. All of his muscles are just relaxed. You can't send a nervous signal to contract any of your muscles. Then Clostridioides. This used to be called a Clostridium, although the more it was studied, the more we realized it was very different from other Clostridiums, so it formed its own genus, Clostridioides. These are still rod-shaped, endospore-producing, and obligate anaerobes. One famous example of this is C. difficile, which you may have heard of. It causes a serious diarrhea. It's related to overuse of antibiotics, and as those antibiotics are overused, it is related to the normal microbiome, those bacteria being killed off, those healthy bacteria being killed off, and then that just allows C. difficile to grow as an opportunistic pathogen in that area. Then Bacillus. Bacillus, as its name implies, is also rod-shaped and endospore producing. This bacteria is common in soil. You probably come in contact with it a lot. We used Bacillus subtilis in lab. Nice thing about Bacillus, very few are pathogenic. So most bacilli are not going to harm you. However, two famous examples. One is Bacillus anthracis, which causes anthrax. This can cause skin infections like you see here. It can also cause pulmonary or lung infections and GI or food gastrointestinal related issues. And then Bacillus cereus. Cereus sounds like cereal to me, so I always remember this is a food poisoning. Although it's not carried on cereal, so don't let that mnemonic confuse you. But Bacillus cereus can cause a form of food poisoning. Then Staphylococcus. You're very comfortable with this at this point. We've used it a lot in lab. Its name implies it is in clusters of balls. It's spherical clusters. Staphylococcus aureus. That name aureus comes from the word for gold because these bacteria naturally by themselves have a slight yellow pigment to them. Staph aureus grows on the skin of a lot of us. About a third of us naturally carry it. And so sometimes it can infect things that have been touched a lot. One important implication of that would be surgical wounds. And it can then produce an enterotoxin, which leads to vomiting and nausea. One very important thing about Staph aureus we've been talking about all year is its quick ability to develop resistance to antibiotics. So now we have MRSA, which is methicillin-resistant Staph aureus, and VRSA, which is vancomycin-resistant Staph aureus. So these are bacteria that are quickly becoming difficult to treat with any known antibiotics. And even as we develop new antibiotics, it still may be able to develop resistance. Then Lactobacilli. These are air-tolerant anaerobes. They don't care if oxygen is present or not. It is part of our normal microbiome. In humans, it's found in the vagina, the intestinal tract, and the oral cavity. It's important in those areas. It produces acid that helps prevent the overgrowth of yeast infection. And commercially, it's used to ferment foods. It can produce lactic acid fermentation, which means it makes lactic acid as it is breaking down sugars as their food source. So it's used to make sauerkraut, pickles, buttermilk, and yogurt. Listeria, one species within the genus of Listeria is Listeria monocytogenes, which is a pathogen. It's a facultative anaerobe that can contaminate dairy and cause a disease known as listeriosis. And it can still grow in the refrigerator. So if your dairy is contaminated, you refrigerate it. That doesn't stop it from growing. You still are at risk for that. It mainly affects immunocompromised patients. Most people that have a functional immune system are going to be fine. It also can cause miscarriage. And so this is the reason, the risk of listeriosis is the reason that pregnant women are told to avoid eating soft cheeses, because if this is in their food, it could harm the baby. And Listeria is also able to survive within phagocytic cells. Streptococcus is a bacteria that's spherical, so you can see it is round, and it grows in chains. So we have these trains of them growing here. We've already talked about its pathogenicity in chapter 15, but let's just review. It's able to destroy phagocytic cells. It makes enzymes that destroy connective tissue and destroy blood clots, so it's able to spread through the body. It leads to strep throat, meningitis, which is an infection of the meninges, pneumonia, which is an infection of the lungs, and endocarditis, which is a heart infection. Not too long ago, I had a heart surgery for one of my heart valves was faulty, and within my recovery group, there were a couple of people that had endocarditis caused by strep throat. So caused by streptococcus. So even though this is something that you can prevent when it's at strep throat, treat it, it can still spread. There are still cases of meningitis, pneumonia, and endocarditis caused by streptococcus, even in modern times. Then Enterococcus, this is a bacteria that used to be grouped as a Streptococcus, but now has its own genus. It is one that we've used in lab. Enterococcus is good at growing in parts of the body that have a lot of nutrients, but not a lot of oxygen. So this would mainly include the GI tract, the vagina, and the oral cavity. You don't hear a lot of cases of these infections unless it's in a nosocomial setting, within a hospital setting. And it is a leading cause of hospital-acquired infections because it does have a high resistance to antibiotics, and it is able to infect surgical wounds, and it can infect the urinary tract through, improper procedures for catheter use. Two species. One is Enterococcus faecalis, which we have used in lab, and one is Enterococcus faecium. Then mycoplasmas. We've talked about mycoplasmas briefly before. I hope you remember, but if you don't, these are bacteria that do not have a cell wall. So that name plasma on it reminds me they have a plasma membrane, and that's it. There's no cell wall outside of that. Because they don't have a cell wall, they don't have peptidoglycan, they're going to be unaffected by a lot of common antibiotics. So penicillin is not going to do anything to this. To still give it some osmotic pressure, it does have sterols in their cell membrane. And without having a cell wall, they're able to be a little bit more flexible in shape, or pleomorphic. Do you remember that word? Pleomorphic means they can take various shapes. And so they can form these filaments that kind of look like fungi. So, this is a colony of bacteria, but it does look like a mold growth here, doesn't it? So that's where that myco part of their name came from. So mycoplasmas are bacteria that just kind of look like fungi because they don't have a cell wall. That's what mycoplasma means. One species within the Mycoplasma genus is Mycoplasma pneumonia, which can cause a mild form of pneumonia. Okay, I will meet you back in our next video where we'll start talking about actinobacteria. See you later. Bye.